Okay, polypeptides. We'll call these proteins. This is an example of a biopolymer. And all polymers consist of monomers. So what are the monomers when you think of proteins as biopolymers? They are amino acids. So let's draw a few amino acids. And when I say a few, I mean 3, and I'm going to draw them kind of in an alternating fashion because it's going to more easily map to what our final protein looks like. So these are our amino acids. They're going to be the monomers from which we construct our polymer. Proteins are condensation polymers. And so, it's not surprising we see acid derivatives in here. We see carboxylic acids. And these carboxylic acids are going to react with amines to form new amide bonds. And so here going across the screen are our monomers, our amino acids. And we haven't specified what amino acid we have. We just put in R groups. Some kind of R groups. These are glycines, alanines, phenylalanines, tyrosines, you name it. It could be anything. So when these react and when they're brought together in the body, they're going to connect and we will release a mole of water or a molecule of water for every condensation we do. So we're going to end up losing this. We're going to go from one acid derivative, the carboxylic acid, to a new acid derivative, an amide. And that's connected our first monomer to our second monomer. And now our second monomer will connect to the third monomer. Again, we will lose water. And this isn't a mechanism. We're just lassoing off water. And that forms our new, that's not an amide bond. That's a carbon-nitrogen bond. Here is our new amide bond that has formed. We can call these amide linkages. They define what we call then the backbone of this protein, as I kind of bold in these sigma bonds. Now we've only made 2 amide linkages, but you can imagine this chain continuing in this direction and in this direction. This whole process is exquisitely controlled by the body's machinery. And this is where we get into messenger RNA and tRNA. And all these tRNAs individually, they're huge molecules, these big tRNAs. tRNA, if you don't recognize that picture. And they contain, each tRNA holds one amino acid. And as these tRNAs stack together against each other, and these are supposed to be tRNAs, they control which amino acids in the order. And of course, down here is our messenger RNA. And that makes sure that our tRNAs line up in the correct order. I mean, it's an incredibly complex process. But the complexity of the process allows us to create these proteins with great specificity. We get exactly the protein we want with exactly the correct amino acids in the chain. And this is something that we really can't duplicate in a synthetic polymer. We can't control exactly the order of monomers in this fashion. So this is part of the beauty of biopolymers. All this machinery, these enzymes, help us create exactly the protein we want. But polypeptides, these are condensation polymers. We can make these condensation polymers. We can also reverse this reaction in the middle of the screen. We can use proteases to break down these proteins. So we can basically recycle these monomers and create all completely new proteins after we break down another protein. So it's an incredibly efficient, exquisitely designed process.